Welcome everyone. I'm Sondhi, a PhD student at Florida International University. I'm going to tell you about how light environment has driven the evolution of visual genes in butterflies and moths. This work was done in collaboration with the Florida Museum of Natural History under the supervision of Dr. Jamie Theobald and Dr. Akito Kawahara. A small disclaimer, this work is in press at Communications Biology. To access the preprint on bioRxiv, please scan the QR code or use the link below. I would like to thank the team that has made this project possible, including my advisors and our collaborators Dr. Bybee and Dr. Ellis. Light levels in nature vary dramatically over time and space. The figure here shows how light intensity changes over the course of a day. We can see that light levels drop almost 1,000 times as you go from a bright sunny day to a twilight dusk, and almost 1,000,000 times when compared to a moonlit night. Yet, it is remarkable that animal eyes and visual systems adapt and continue seeing the world over this vast range of light intensities. Animals have to adapt to changing light intensity while successfully foraging, finding mates, and escaping predators. Dim light poses more challenges than bright light, yet offers many advantages such as reduced predation, and animals have adapted to function in extremely low light environments. This figure shows the dimmest light level different organisms can see color. A diurnal hawk moth can see color up till dawn or dusk. As humans, we can see color under moonlight, and the nocturnal elephant hawk moth can see color under starlight, which is almost 1,000 times dimmer. Similarly, some butterflies have been shown to have excellent color discrimination ability with many more light sensors than humans. But with more than 150,000 butterfly and moth species, we know a little bit about the variation in their visual systems. Butterflies and moths also show considerable variation across the tree of life in dia niche, which is the time of day each species prefers to be active. The figure below is an adaptation of a phylogeny of butterflies and moths published in PNAS by Kawara et al. 2019. We identify the dia niche of different species in their phylogeny by consulting with experts, literature, and natural history databases. And in this figure, yellow shows diurnal or day active species, blue shows nocturnal or night active species, and green are species that are active both during the night and the day. You can see that while butterflies in yellow are more diurnal and moths are more nocturnal, there are many examples of species that have switched their DL niche across the tree. We decided to investigate how this variation in DL niche might affect visual system evolution. We focused on visual genes since they have not been widely studied across the tree, and including this recent paper, there were over 175 transcriptomes and genomes of Lepidoptera available. I will walk you through the three lines of evidence we found for how light environment has influenced visual gene evolution. We looked at the changes in the number of visual genes, the evolution of protein sequence, and how that affected the 3D protein structure and function. First, we looked at how many visual genes we could find in each species. Little background is necessary. On the left is a model of the photopigment opsin. It is composed of a transmembrane protein in complex with a small light-sensing molecule known as retinal. This molecule absorbs light, changes conformation, and starts a signaling cascade, which results in the transduction of light into an electrical signal. Opsins are used across animals to sense light. However, the opsin protein structure determines which wavelength of light retinal is most sensitive to. And to the right, the three commonly used wavelengths that insects respond to, which is UV, blue, and long wavelength or green, are shown. And correspondingly, the different wavelengths which those options respond to. The combinatorial response of different options is what makes up the foundation of color vision. Insects usually have these three options, but some insects, such as butterflies, can see red or longer wavelength light. Changing the number of options can alter the range of colors the visual system perceives. To the left is the depiction of the three option system that we normally have and how a butterfly appears to such a system. To the top right is what a colorblind person would see. Um, and to the bottom right, is what a dichromat or a partial colorblind person um, would see the world as, or the colors they would represent to them. True colorblind or monochromats are very rare, and more often people have at least two functional options. For example, red-green colorblindness. They can see a reduced dichromatic color space, and thus reducing the number of options can directly modify vision. Similarly, adding more options improves color vision, although models suggest that adding more than four options shows diminishing improvements. Our first task was to identify these options from the genomes and transcriptomes, and we used phylogenetically informed annotation, or PIA, a tool developed by the Dan Spicer lab and the Oakley lab. The figure is an example of the option gene tree generated by PIA with the UV option expanded. The identity of genes is confirmed by their clustering with the reference sequences shown in red. We repeated this for all species and mapped option recovery across the tree. The figure shown here is the same phylogeny of butterflies and moths, color-coded by DL niche, yellow diurnal, blue nocturnal, and green both, but with additionally, the presence or absence of different visual options is marked. Purple, blue, and green refer to three color options, and red is RH7, an option not involved in color vision. Duplications are marked in red, and the number of in the boxes indicate the number of options. To the left is an expanded view of the tree, with some of the species duplication shown to the right. Interestingly, we found 18 duplications across the tree that were almost all in diurnal taxa, or at least in taxa partially active in bright environments. This suggests that duplications are more often maintained in bright environment, likely because they became functional. We also wanted to see if the opsin protein was evolving in cases where there were no duplications, so we examined sequence level evolution. Opsin gene or loss or gain can change color vision, but so can the actual sequence of the opsins. We looked at the rate of synonymous mutations. Mutation on the left, where changing the DNA base does not change the translated amino acid, versus non-synonymous mutations, where a change in the DNA base also changes which amino acid that codon codes for. The ratio of non-synonymous changes to synonymous changes is often called the DN/DS ratio, or the rate of selection, since a value greater than 1 indicates rapid changes, but a value of 0 indicates the protein was not changed as much. We found that if we compare this rate between opsins from nocturnal and diurnal species, the rate was higher for opsins in diurnal species, indicating there were more changes in diurnal opsins. And this was promising, so we tried to look at which amino acids were changing faster and where they were in the protein structure. To the left is a representation of the opsin structure with different amino acids color-coded. The sites surrounding retinal or the photopigment are in yellow. The sites that were under higher selection are in red, and the sites that were in both categories or interacted between both are in pink. You can see the proximity with the yellow and pink sites, but since these amino acids interact in 3D space, we needed 3D models. We used the 3D squid opsin structure, which was the closest structure available, as a basis for a model of the Lepidoptera opsin, which is shown to the left, and mapped the location of the amino acids under high selection to the opsin. We found that many of the positively selected amino acids contacted the amino acids that were bound to retinal and thus could tune the opsin by changing the chemical environment around the chromophore and altering color vision in diurnal species. Thus, using three lines of evidence, which is first, the higher prevalence of duplications in diurnal species, second, the faster rate of diurnal opsin evolution, and third, amino acid under selection capable of tuning color vision genes, we show that light environment drives visual gene evolution across butterflies and moths. These findings are very interesting because they show that While color vision is important, it is possibly more important in diurnal species than nocturnal species, and there is possibly something that prevents nocturnal species from diversifying their color vision, but that has allowed diurnal species to do so. Our future directions include looking at the gene expression, the experimental validation of some of these mutations, and exploring the behavioral morphology that accompanies DL niche transitions. I would like to thank funding agencies and our colleagues who supported this project. Thank you.